Well, I'm going to get started. Why not? I'm Ed Levin, Director of the Duke Center on Addiction and Behavior Change, and welcome to our seminar series. We're pretty far along in this fall 2021 semester seminar schedule. And before I introduce today's speaker, I just want to look ahead to the next couple of talks. First, I want to wish everybody happy Thanksgiving. This is our last meeting before Thanksgiving, and then we'll pick it up right after that, December 1st, with Andrew Hockey from the psychiatry department here at Duke, talking about smoking risks beyond nicotine. Certainly, tobacco smoke has 4,000 or more compounds in it, some of which are quite neuroactive and neurotoxic. So he'll be looking at PAH toxicity with primarily benzopyrene. And Connor O'Neill will then follow on a week after that, talking about evidence-based treatment, co-occurring substance use in adolescents and adults. And he is also from the psychiatry department, talking about his clinical work. And then that'll be it for the fall semester. And then we're going to pick it up in the spring, starting January 19th with Linda Perino from Wake Forest. And she's going to be talking about her career, very active career, and consequences of chronic drug use and her overview of that. And then importantly, later in January, we're going to have participants and graduates of the TROSA program. So that is, they'll be talking about real life recovery from a drug addiction. And if you're not familiar with TROSA, please get familiar. And this will be a good chance to get acquainted with the people and the program there. And it's really spectacular in terms of helping people overcome addiction. And then we'll have a whole series of talks, but I just wanted to highlight on February 22nd, we're working together with Dr. Wanda Boone from Durham Tri, looking, and we'll have a symposium on marijuana, brain science, and community impact for risk prevention and legalization. So here is the description of that. And all of these are on the CABC website and on the DIBS website. So please go ahead and do that. And we're going to be having registration for the symposium. It's free for all comers, but we would like to get people to register for that. And that link is on the website. Okay, so now it's my honor and privilege to introduce Dr. S. Alex Marshall, Assistant Professor of Biological and Biomedical Sciences at North Carolina Central University. Alex earned his bachelor's in zoology at the University of Florida and PhD in pharmaceutical sciences from the University of Kentucky in 2013, and is now on the faculty of NCCU. And his lab is primarily focused on understanding neuronal mechanisms and they contribute to and result from alcohol abuse, and in particular looking at immune systems and their interactions with neural systems and adolescent alcohol abuse in animal models. So today he's going to speak to us about the neuroimmune response as a mediator and consequence of alcohol abuse. Alex?
I did that rookie thing of not unmuting myself. Thank you so much for the invitation. I'm excited to be here to talk about my work on the neuroimmune response and its relationship to alcohol. Before I get started, I just want to, another way that I often phrase this, which I can't take credit for, it came from Dr. Cynthia Kane at UAMS, was we talk about the reciprocal reinforcing relationship between alcohol abuse and the neuroimmune system, because she made fun of me about my alliteration and my need to use poetic terms at times. But essentially, that's what I'm going to be talking about. So we're going to go back and forth between the biological and the behavioral aspects of the neuroimmune system. So just as an outline, I'm going to be really brief in terms of alcohol abuse, considering the group. I'm not as familiar or certain that everybody knows about the neuroimmune response, so I'm going to give a very crude overview of how we study it. We particularly focus on two different regions of the brain, and then I'm going to talk about the neuroimmune response as a consequence of alcohol and maybe as a mediator. So how does it actually impact the behavioral aspects? Within that, our lab is also interested in both binge consumption, so early binge consumption and non-dependent situations, as well as an alcohol dependence and how it affects neurodegeneration. So as you all are very aware, alcohol use disorder is characterized or classified as a disease state. It's been designated as such by both the American Psychological Association and the World Health Organization. And some of the aspects that are associated with being diagnosed as having an alcohol use disorder include a loss of control. So you drink more than you intend, or you drink when you don't intend to. Preoccupation with the drug. So you drink when you're supposed to be doing other things. Maybe you're supposed to be doing coursework. Maybe you're supposed to be at somebody's social event or party. And then lastly, the more biological signs of an alcohol use disorder include the development of tolerance and withdrawal. And so I put this slide in here. I can't also take credit for this. This is Sean Ferris's slide, but I liked it so much that I was like, hey, let's all use this. But to indicate that what I'm studying is a subset of people who use alcohol. After I give these talks, every time somebody's like, oh, so I shouldn't drink at all. Like, you said that it impacts all these things. What I'm talking about is binge consumption and alcohol dependence. And so what this diagram is setting up is that there is a third of the population that doesn't drink. Sorry, you can't see my pointer. There's about a third of the population that doesn't drink at all. A third that you would say, a third to maybe half that you would say is just consuming alcohol in social events. And what we're most interested in is this top 9th and 10th percent. So the top 9th, the 9th and 10th decile. And so there's a group of people who, and this is drinks per week, I should say, who are drinking well over 73 to 74 drinks in a week. So this would be those who would be classified as maybe being alcohol dependent or have the likelihood of having alcohol-induced brain damage, as opposed to the ninth decile, which would maybe be those who are binge drinking problematically. And so just to put it into a little bit more text, about 5% of the world is considered to have an alcohol use disorder. And I should change that wording because we are no longer saying things like addicted. I need to change that to have an alcohol use disorder. In the United States, that means there's about 20 million people. And for those who maybe don't drink or aren't interested in numbers, maybe you're more interested in the money that it costs. Some of the financial burden associated with alcohol use disorders, whether it is from health problems or loss of missing work or property damage, costs the United States approximately $250 billion a year. Of those individuals with alcohol use disorders, even less choose to seek treatment. So what this is showing is that less than 20% of people actually seek treatment. And of those that are seeking treatment, an even smaller percentage seek medical or use a medication as a part of their treatment. So one of the things that I'm interested in terms of understanding the neuroimmune response to alcohol, as well as how it mediates alcohol, is whether or not the neuroimmune system could potentially be a site or a target for the treatment of alcohol abuse. And so as I alluded to at the very beginning, my lab is kind of split. We study alcohol abuse, or what was formerly known as alcohol abuse, which would be like binge intoxication and binge drinking. But we also study alcohol dependence or alcohol-induced brain damage. Along those lines, I guess this is missing an N. Along those lines, there are changes in the system in terms of initially people often binge drink and experiment with alcohol. When you continuously binge drink, there are neural adaptations or maladaptations that occur that can change tolerance, can change your perception of the drug, which leads to withdrawal. And importantly, in terms of how it affects neurodegeneration, it leads to deficits in executive function, which then potentiates the cycle of alcohol abuse. So I say all that just to say that the term alcohol use disorder is a broad continuum of people that are binge drinking and drinking problematically, and those that would be previously be considered alcohol dependent. So within the many maladaptations that occur in response to alcohol, my lab predominantly focuses on the neuroimmune system. And so I just wanted to briefly introduce what I mean by the neuroimmune system. And so for the purposes of today, I'm gonna only talk about it as it relates to glia and cytokines. And so the glial cells have the ability to secrete cytokines and chemokines that can lead to or increase the peripheral immune response or the recruitment of the peripheral immune response. And so what this diagram is setting up is that there are these cells called microglia, which can hone in when there's damage, and they have the ability to phagocytose damaged neurons or damaged cells in the brain. But their response is often mediated by cytokines and chemokines that are secreted by astrocytes. So in the lab, we go back and forth between studying microglia and astrocytes, as well as these chemical transmitters or cytokines that they secrete. In general, much like the peripheral immune response, the neuroimmune response is thought to be associated with protection and recovery. So it protects the body from infections, but also after an infection or after a traumatic brain injury, it's responsible for recovery. So it can also secrete trophic factors that say, hey, neuron, I know that you aren't feeling well. Here's a little bit of BDNF. I hope it makes you feel better. Finally, one of the things that it's come out of this neuroimmune response is the idea of neuromodulation, with the idea that these cytokines can actually change neurotransmission, and I'll talk about that in a few more slides. As it relates to the neuroimmune response, again, in terms of the classical neuroimmune response in protection and recovery, it's thought that the microglia and astrocytes are activated as in response to a pathogen. And so that pathogen can be bacteria. It can be, in this case, this is showing beta amyloid. So it can be aggregates of proteins that are in the wrong location. And that glial activation includes the secretion of these cytokines and the activation of these cells to get rid of those foreign invaders. In a classical neuroinflammatory response, you also get disruption of the blood-brain barrier. And so what this is showing is disruption of those endothelial cells with neutrophils entering into the CNS. And so often when you hear somebody say neuroimmune response, it is technically different from a neuroinflammatory response. So the neuroimmune response participates in neuroinflammation, but neuroinflammation implies that you have the peripheral immune response involved. So why do we care about the neuroimmune response as it relates to alcohol abuse? So actually right when I was starting grad school, so this says 2008, so that's when I started grad school, Fulton Cruz at UNC, as well as some others, put out some papers showing that, again, I have these old terms in here. I'm gonna get in trouble if George Koop sees this, but people with alcohol use disorders have evidence postmortemly of an activated immune response. So on the left, what I'm showing is IBA1. IBA1 is a marker for microglia that I'll show over and over in this particular slide deck. But there's a control person who might have been a social drinker versus someone who would be considered alcohol dependent. And you can see that there are clear differences between the microglia in terms of their morphology, and that morphological change is thought to be indicative of activation. In congruence with in agreement with this microglial change, they saw changes in chemokines like MCP1. So this is all in human alcoholics or in clinical populations. So you get increases in the chemokines that are secreted by activated microglia in different subregions of the brain. as well as the receptors that will respond to and mediate the immune response. So we're showing 3 different ways that the immune response has been changed in clinical settings for alcoholics. The original hypothesis behind the implications of that neuroimmune response was that it was associated with neurodegeneration. So as I alluded to at the beginning, when you have a neuroimmune response, it can engulf neurons or engulf damaged cells. But if you have an overactivation of the system, you're going to remove healthy tissue. And so what has been previously shown over and over is that alcohol use disorders can cause neurodegeneration. And so that neurodegeneration, some people will classify an alcohol use disorder as a neurodegenerative disease. But whether or not you agree with that, it is clear that there are changes in the ventricles and the gyri in terms of their size, as well as in the general white and gray matter of a social drinker versus an alcoholic. However, when I was in grad school, one of the things that I got interested in was what other impacts does the neuroimmune system have? And so this is where I want to talk about neuromodulation a little bit. And so the majority of this comes out of the general malaise associated with sickness. And so Dr. Kelly at, I think he was in Illinois or Indiana, one of those I schools, did a lot of work discussing how the peripheral immune response actually can be associated with general malaise / depression and just the general feeling of not feeling well. But within that, what they discovered was that these neurons actually have receptors that can respond to what were initially thought to only be involved with the neuroinflammatory response. So the cytokines that are secreted by these activated glia can directly impact neurons. And the only one that I want to focus on, this one is from a pain study showing that these cytokines and chemokines can impact pain. But the one that I want to focus in on the most is the idea that the pro-inflammatory response, IL-1 beta and IL-6 or pro-inflammatory cytokines, can inhibit GABA and increase glutamatergic signaling. When it comes to alcohol pharmacology, as you all are aware, it has the opposite effect. So if you have an increase in this inflammatory signaling, it's actually combating ethanol's effects. And so one of the things that we have seen, which I'll show in a few moments, is that alcohol can change these cytokines. And so it's possible that targeting these cytokines might impact ethanol's pharmacology. So getting back to why we study it, I think that everybody that's on recognizes that one of the goals for this is to find a treatment. And so the current treatments for alcohol use disorders seem to be very specific for different groups. So acamprosate seems to be a target for those who have craving or to combat craving, as opposed to naltrexone, which is more associated with inhibiting reward from alcohol. But what's interesting about potentially targeting the neuroimmune response, and the reason that my lab is set up the way it is that targeting the neuroimmune response has the ability to address both the behavioral problems associated with alcohol abuse, as well as the biological change in terms of neurodegeneration. So of these 3 drugs, while they can, and they do correct some of the alcohol use disorders, none of them are associated with the reversal of the damaging effects of alcohol. And so the last intro slide is that, again, I don't have to say this for this group, but we're gonna toggle back and forth between hippocampal and amygdala data, with the idea that both are involved with alcohol-related issues. So my first question is, what happens to the neuroimmune response? What is the consequence of alcohol use disorders, binge drinking, as it relates to these neuroimmune responses. And so in order to study that, we use the drinking in the dark model. And so the drinking in the dark model is a model of excessive binge-like consumption, where we allow the animals to have access to freely consume alcohol during a limited period during the dark. So for the first 3 days, they have 2 hours of access. And on day 4, depending on the study, they might have 2 or 4 hours. So this 4-day period is considered one cycle. And we also increase the number of cycles depending on the study. The drink in the dark model has been repeatedly shown to elicit ethanol consumption that is comparable to binge-like levels. So we get BDC somewhere around 100 to 120. And their consumption is considered voluntary depending on who's reviewing the paper, because they're not drinking the alcohol for its caloric properties. They're drinking the alcohol because, or our assumption is that they're drinking the alcohol because they are getting the pharmacologic effects. In order to characterize the response, we use very classical tools. So we use PCR to look at mRNA. And so you'll see data that says that we gave them ethanol and then we looked multiple days out. And one of the reasons that we do that is we want to understand the persisting effects. So we give them alcohol, we might look at it immediately, 7 days, sometimes even 28 days after abstinence. And the other thing that our lab does a lot is to look at the threshold that changes. So when does binge drinking change the threshold? Is it after one bout of the cycle, after 3, or even 10 cycles of the drinking in the dark paradigm? So one of the first studies that I did after within the drinking in the dark model was to look at pro-inflammatory cytokines. And so what this is showing is that in the basolateral amygdala, but not in the central amygdala, we saw an increase in IL-1 beta. While the data is suggestive that it happens after one cycle, it was not significantly different until after 3 cycles. So that was specific to the basolateral amygdala and didn't occur when we gave sucrose. We then went on to look at IL-10, an anti-inflammatory cytokine. And so when you see an increase in a pro-inflammatory cytokine, the seesaw balance or the homeostatic response is to decrease the synthesis of anti-inflammatory cytokines, and we saw exactly that. So at 3 weeks in the basolateral amygdala, but again, not in the central amygdala, we saw a decrease in IL-10 immunoreactivity. And again, that seemed to be specific to ethanol and not occur in our sucrose animals. The change in cytokines was also specific to the CNS. So we did ELISAs, or we used ELISAs to look at IL-10 specifically in the amygdala and the serum samples. And again, only in the amygdala was there a decrease in IL-10, but this didn't occur in the serum, suggesting that early, I say early, but binge-like consumption or non-dependence consumption is more susceptible, the brain is more susceptible than the peripheral system to this inflammatory response. So those were the cytokines that we looked at. We've looked at a couple others that I didn't include, but we also look at the glial responses. And so what this is showing is changes in glial gene expression. And this was done by one of the first students in my lab, Bella Grafassi. Sorry, I paused on her name. But what she found was that there was an increase immediately after alcohol consumption in AIF1, which is a gene that should be present in all microglia, as opposed to ITGAM, which is upregulated when the microglia are activated. But it was only immediately after alcohol consumption. And after just 1 day and 10 days, there was no significant difference between that and the controls. We went on to use immunohistochemistry to look at the actual morphology and some of the markers associated with the increase in microglial activation that we saw at the gene level. And so what this is showing is OX42, which is a marker for CD11B. OX42 is thought to be more specific to an immunological response. And so whether it is pro or anti-inflammatory is kind of debatable, but it at least says that there's been an immunological response by the microglia in a recent history. And what we found was after both 1 and 3 cycles of ethanol, there was an increase specifically in the dentate gyrus. So it seems to be in specific regions. We didn't see anything in the other subregions of the hippocampus. And one of the things that I think everybody gets confused about, and I keep meaning to change this when I present this, is that the CA1 and CA2-3 regions of the hippocampus actually have much lower microglia density than the dentate gyrus. So this is showing 4% as opposed to 0.6, just for clarity. And this was done by the lab manager and the lab J. Nelson. Going back to other glial cell types, we've done the same thing looking at astrocytes. And so this is one of the first papers that Bella put out. So what she found again was that it seemed to be specific to 3 weeks of ethanol consumption, so that there seems to be a switch after 3 cycles of this paradigm where you get an increase in the immunoreactivity for GFAP. And there was no change after 1 cycle. In particular, this seemed to occur in the dentate gyrus and in the CA1 region, but not in the CA2-3 subregion. My lab actually carries on with these characterization kind of experiments to look at other variables. And so one of the variables that we look at quite often is sex differences. So this was another student that originally was in my lab at High Point, Chris, who looked at the sex differences in regards to the astrocytic response. And again, what we found was that ethanol obviously increases the percent or densitometry of the astrocytes, but the response seemed to be greater in the female animals compared to the males. So if we do like a percent control, there's actually both a basal change in terms of females having more astrocytes, regardless astrocyte density in the dentate gyrus, regardless of ethanol. But in the presence of ethanol, it seems to actually be a greater response than that which is present in the males. Likewise, we also do a lot of things when it comes to aging. And so Bella has also done some work looking at aged animals. And so we do young versus aged. And one of the things that we found, which is particularly interesting to me in terms of the alcohol effects on the neuroimmune system, is that binge drinking almost should cause an immune response. So you have this toxin that is present, that has the ability to kill cells, not necessarily at the levels that we're giving it, but you should have a kind of a neuroimmune response. So the IBA1 or the microglia are reactive in the younger animals, but in the aged population, because they have a more robust density of the microglia, it's actually less responsive. So the senescent response, or this lack of response, could actually indicate that it is more problematic to drink as you're aging, and that the neuroimmune response might play a role in that. So that is the consequence of alcohol in terms of the neuroimmune response. Now I want to talk about how the neuroimmune response can modulate the alcohol system. So what is this reciprocal relationship? So we're not the first ones to do this by any stretch of the imagination. There's been a variety of studies that have shown that immunomodulators, including PDE4 inhibitors, which I'm showing here, like Periph and I'm not going to say that right, Mesopram, can reduce ethanol consumption in the preclinical studies. And so this is actually out of Texas. Yuri Blednov in Texas has done a lot of work looking at a variety of NSAIDs and immunomodulators that reduce binge drinking. They've also done a lot of clinical work showing that things like ibutylast can actually reduce craving, as well as the rewarding effects of alcohol. So it reduces craving. Ibutylast reduces craving compared to placebo. And after consumption, how much they liked the ethanol. I just read that the ibutylast is actually apparently in a phase 2 clinical trial to be used for alcohol use disorders. So it was a reminder that I need to stay up on the literature a little bit. But those drugs are given peripherally. One of the interests in my lab is to probe the neural circuitry. And so in order to do that, we not only target more specific components of the neuroimmune response. So those PDE4 inhibitors and ibutylast are kind of general neuroimmune modulators. We wanted to look more specifically to determine which cytokines and which cells are actually mediating these responses, as well as which subregions. And so in order to do that, we use stereotactic surgery. And in this case, we're still talking about the drinking in the dark model. We allow them to have or to consume alcohol to get a baseline of their consumption. They undergo surgery, and then we use a Latin square design to determine how much the manipulation impacts their consumption. And so following up on our increase in IL-1 beta that I showed at the very beginning was a consequence of alcohol, we wanted to look at IL-1 receptor antagonists, or IL-1RA, to combat that effect. So we gave IL-1RA directly into the basolateral amygdala. And what this is showing is that it decreased ethanol consumption. And importantly, for our work, it decreased ethanol consumption sufficiently enough to fall below the binge drinking level. So binge drinking is considered 80 milligrams per deciliter, and these guys have a blood ethanol concentration around 40 to 45. It did seem to be specific to the basolateral amygdala, or at least the dose that we used seemed to be specific to the BLA. So when we gave the IL-1 receptor antagonist into the CEA, there were no changes in either consumption or blood ethanol concentrations. Whenever you do this kind of work, you want to follow up with behavioral controls. And so we used those same animals and gave them IL-1 receptor antagonists and looked to see their sucrose consumption. There was no change in sucrose consumption, suggesting that the IL-1 receptor antagonist is not general to all reward, but seemed to be specific to ethanol. We also followed up to make sure that our IL-1 receptor antagonist was not changing motor activity or anxiety-like behaviors using the open field test. And there were no changes in the total distance traveled or in their locomotor activity or in any measures that are considered to be indicative of anxiety-like behaviors. So not only are we interested in the messengers that mediate the immune response, we also want to manipulate the particular glial cells. So designer receptor exclusively activated by designer drugs or DREDs can be targeted to either be incorporated specifically in astrocytes or in microglia. And so in the work that I'm going to show today, I'm only going to talk about the astrocytic work. But as you all know, giving this DRED, it's incorporated into the astrocytes, and we can give CNO to either activate or inhibit the astrocytic G protein couple receptor signaling. And I say it that way because for those that use DREDs in neurons, it is important to recognize that while the HN3DQ, GQ activation, and GI activation is also often talked about as being excitatory or inhibitory, it actually turns out that the neuroimmune response or the astrocytic response is much more calibrated so that the GI and GQ response can actually have similar changes. So it's not that GQ is pro-inflammatory or anti-inflammatory. And I just wanted to set that up before we get to the next slide, okay? So if you assume that GQ activation would be pro-inflammatory, GQ signaling would, you would think that GQ activation would increase ethanol consumption based on what we've been showing so far. But we do have some preliminary data, which I'm not going to show today, that suggests that GQ signaling and the astrocytes under these conditions actually inhibits or leads to an increase in anti-inflammatory cytokines. But what I'm showing, at least today, is that it decreases ethanol consumption. So we had these transgenic animals that expressed astrocytes throughout the system. And when we gave them CNO on the last day of the cycle, so this is the first 3 days where they got saline, and on the last day they got the CNO. And when they got the CNO, only in the GQ dread positive animals was there a decrease in consumption. The CNO had no independent effect on the wild type C57 mice or in the litter mate controls from our breeding colony. Unfortunately, what we figured out was that GQ signaling and astrocytes apparently affected everything if you put it throughout the system. So it actually will be incorporated both in the CNS and in the peripheral nervous system. But we looked at sucrose, and CNO decreased sucrose. And then we looked at their travel time, and CNO actually impacted locomotor activity. So the thought is that the GQ activation, when it is universal or throughout the system, impacts locomotor activity. And so the GQ target may not be as effective, but we followed up those studies and injected CNO directly into the basolateral amygdala. And what we found is that when you are more specific about where the CNO is administered, therefore more specific about where the astrocytes are, or GQ signaling, and the astrocytes is activated, it seemed to be specific to ethanol. So what this diagram is showing is ethanol consumption. And we actually did multiple tests. I'm only showing the first two. And each time they received CNO, there was a slight decrease in their ethanol consumption that was also consistent with a decrease in their blood ethanol concentrations. One of the things that we did find out is that this response seems to be predominantly mediated by the first 2 hours. And so we are actually going back to repeat these studies using different timelines. But importantly, there was no change in sucrose consumption. So regardless of whether the animal got vehicle or CNO, they consumed the same amount of sucrose throughout the experiment. All of this says that the neuroimmune response can occur as it relates to alcohol abuse, independent of brain damage, and alludes to the fact that the neuroimmune response can directly alter ethanol consumption. And so in the last, hopefully just 10 minutes, I want to talk about the second kind of component of the lab, which is looking at the consequences and mediation associated with alcohol-related brain damage. And so going back to the idea of the consequence, first I want to show how does alcohol dependence and alcohol-induced neurodegeneration affect neuroimmune responses, and then I'll show a little bit of data about how we manipulated the neuroimmune response. And so in order to do this, the first line of work that I'm showing uses a rat model called the Macrowitz model, where the animals are administered ethanol 3 times a day for 4 days by intragastric gavage. When they do this, they reach BECs well over 300 to 400 milligrams per deciliter. And I say that just to show the distinction between the models. So we're talking about right at the legal limit between 80 and 120, and then 3 to 4 times the legal limit when we're talking about alcohol-induced brain damage and dependence. And to further clarify the distinction between the models, again, the voluntary binge consumption or the drinking in the dark does not cause an increase in cell death, as opposed to my intragastric gavage model that I'll be showing, where we do see increases in cell death markers, specifically FLOR-JC. So this first set of data that I'm showing is actually left over from my graduate work, where we showed that much like during the during the drinking in the dark phase, when you have alcohol-dependent animals, there is a marked increase in microglia activation. And so this is tritiated PK1195, which is a marker for microglia activation. And what we found is that immediately after ethanol consumption, And even after 7 and 28 days, I'm not showing the 28-day data, that you get this marked increase in microglial activation. I will say at the 28-day mark, it was still increased, but there is some resolution in terms of the going back to normal. Sorry, I don't know why I paused there. So similar to the other studies, looking at glial responses and looking at cytokine responses, what we found is that there was no change in this case in IL-10. So in the other models, when we saw an increase in the inflammatory response, there was a decrease in the anti-inflammatory cytokines. But what we found is that after multiple cycles of ethanol, you do see a marked increase in markers like TNF alpha. We also looked at IL-6 and IL-1 beta, but it's indicative of the hippocampus responding to, or the neuroimmune response responding to ethanol at these high levels. Going back to looking at other factors associated with this, one of the things that we are really interested in is looking at what's called the dystrophic microglia within these aging populations. And so, gosh, I'm forgetting his name. Straight down at Florida has done a lot of work showing that microglia become senescent and dystrophic, or have this beating processes associated with aging. And when we were going through counting all these cells, actually it was when I was going through counting all these cells, we saw these weird morphologies and just kind of followed up to try to understand what was going on. And it led us into this direction that not only can a hyper-inflammatory response that we're seeing with the increase in TNF alpha and microglia activation impact neurodegeneration, but a hypo response or a lack of response or senescence can also cause or be associated with neurodegeneration. And what we found is that both in the young and aged populations, there was an increase in the, a decrease in the number of microglial cells after this high level of ethanol exposure. But importantly, we've shown that there's an increase in the number of dystrophic cells, particularly in the aged populations. As it relates to manipulating the neuroimmune response after an alcohol-related brain damage, I will say that this particular research is much early in its formation than that for the drinking in the dark model. But we've switched a little bit. So instead of using rats, we now use mice, and we're doing the 10-day gavage model. They get a daily 5 gram per kilogram gavage, which is much nicer than coming in 3 times a day to gavage the animals. But at any rate, this model has also been shown to be associated with alcohol-related brain damage and BECs that are between 200 to 300 milligrams per deciliter. And so one of the first studies that we've done that I'm showing here was kind of a null data, but I figured I'd show it anyway. So Maya, one of my first students at North Carolina Central, looked at changes in FLOR-JC or neurodegeneration after alcohol exposure. And so alcohol still increased the number of FLOR-JC positive cells. We gave etifoxine, which is it's a GABA agonist, but has also been shown to impact PK1195. And so we thought that by inhibiting the immune response, we might recuperate or rescue some of this alcohol-related brain damage. And unfortunately, we have not seen that directly for the alcohol-related brain damage. We have seen some evidence that the immunomodulation still occurs. So we've been looking at astrocytes and microglia in these same animals and have seen some influence of etifoxine under those glial responses. So what I've shown today is that the neuroimmune response can happen independent of damage, that the neuroimmune response can alter ethanol consumption. And then in the latter half, I talked about how the glial activation can occur in response to damage, exacerbate damage. And what I didn't talk about today is how that damage leads to cognitive decline. So the take-home message, because this is actually out in my classes with some cliff notes, is to remember that the neuroimmune response has, regardless of the parameters or models that we've used, have shown a robust change in terms of the biological response. Within that, we have gone in and manipulated it and seen that it affects both the biology and the behavior, making the neuroimmune response a potential target for alcohol use disorders. And with that, I still thank my former mentors. So my former mentors from graduate school and my postdoc work, but more importantly, the students that actually did the work that I presented today. So the work that I showed today was mainly from Kayla and Jay, as well as Amaya. But we have a pretty big lab, for me anyway. And I did not actually present any of the work from my collaborators. So my work with Leon Coleman, looking at brain damage, or a different model of brain damage, the adolescent work with Scott, or the work with environmental toxins that I've been doing with Kevin here. And we've been lucky to have some funding. And so with that, I am happy to take any questions and a drink of coffee because I didn't drink the whole time.
Well, thanks for such a great talk, Alex, and I really appreciate all of your work and talking about it. It's so interesting. So I had a question in terms of this immune reaction. Then can you differentiate how much of that comes from the actual bout of intoxication versus the withdrawal from intoxication?
Yeah, so we have actually tried to do some of that. In our dependent model, we have not really successfully been able to knock down withdrawal sufficiently enough to differentiate it. So we didn't want to go as far as giving, like, I guess maybe we should just do like some benzos or something, but we've given things like minocycline and what was it? Dexibuprofen during right before the withdrawal phase to see if it knocks down that response. The minocycline, we didn't get anything, although somebody published and said that they did. And with the dexibuprofen, we were, I think that tissue is probably still somewhere at UK. But the short answer to your question is we attempted it. We did not necessarily see the difference, or we weren't able to knock it down enough to see a difference. But honestly, if I were to just postulate, I think that the majority of it occurs during intoxication, but that the withdrawal response, repeated withdrawal responses, actually potentiates it. So if you go through one bout of withdrawal, I don't think it's as problematic in terms of the responses that we've seen. But there have been people who have done that binge model, the 4-day binge model, like I think they did it 6 times, and they definitely saw a much more robust response because of the multiple withdrawal cycles.
Thanks. Now, do people have questions or comments? Well, yeah, go ahead.
Alex, very nice job. I really liked the flow of that presentation. It was great. You alluded briefly to the interaction of alcohol and aging and inflammation, sort of tripartite interaction there. You know, we know that neuroinflammation is neurotoxic. It changes the way, various cell types interact with one another in the nervous system, and that ethanol and aging both, have components related to neuroinflammation. I wonder if you've thought much about that three-way interaction.
So that is one of my goals. Thus far, we've actually looked at them separately. But one of the things that we have done, which I'm really interested in, and we'll see what happens with it, is we have looked specifically at just aging and manipulated the astrocytes in aging. So we've done our DREAD manipulation to increase GQ activation. One of the things that I did leave out in terms of GQ activation of astrocytes, I focused on the neuroimmune response, but GQ activation can also increase glutamatergic tone with astrocytes. So what we've seen in our aged animals is that it actually might help with some of the learning and memory tests. So we've only done a few so far, but it is definitely something that I'm interested in combining with my alcohol research. Just haven't quite gotten that far yet.
Yeah, no, it's good. I think there's a lot of potential there, and we can certainly talk about it. Aging studies are hard to do because they're
Yeah.
time consuming and all of that, but it's a really important interaction. That alcohol might exacerbate that process, and it might be through an inflammatory mechanism.
Yeah, what I'm gonna hopefully do someday soon is to get a postdoc, because aging studies are definitely not a master's or graduate level project.
Right.
So yes, yes, Dr. Kuhn, sorry.
Yeah.
Hey.
Why the amygdala? I mean, why the BLA?
So our focus on the amygdala, it actually is kind of born out of the relationship between alcohol and stress. And so there's been a lot of studies that have shown that alcohol induces CRF and some of the stress-like peptides in that region, specifically the basolateral and central amygdala. And because the neuroimmune response can also impact CRF, that was originally why we started in the amygdala. So it's kind of whose lab I was in, to be honest. But but that's kind of why we toggle back and forth. So I have an interest in the impact in learning and memory, but also as it relates to stress responses.
Yeah, and if it had been that, you would have seen it more. Probably you would have thought to see it more in the CEA than in the BLA. But I was curious.
So,
it was what it was, though.
Yeah, so Marissa Roberto has actually done quite a bit of work. And one of the things that, we do our work and then we learn more. One of the things that I left out is that I actually think that the reason that we don't see responses in the CEA is probably that we didn't do a dose response. So we chose our dose based on other work. And then probably maybe a year later, Marissa Roberto and I were sitting down and she was like, so we found this in the CEA, but we use this dose. And I was like, okay, that sounds right. So they were able to look at some EPHIS effects of IL-1 receptor antagonism in the basolateral and the central amygdala. So.
Great, that's very helpful. Thank you.
Yes, ma'am.
Okay, other questions?
Yeah, Alex, that was very interesting. And I wanted to sort of dovetail off that last question, because I know with interleukins have been studied for some time as neuroimmune modulators, neuroinflammatory modulators. But I remember a conversation with Stacy Bilbo where we were saying people may want to be open to the possibility that these are general signaling molecules that just happen to be used by the immune system, and that neurons may actually use interleukins as a neuromodulator independent of the inflammatory response.
Yeah, and I would definitely agree with those sentiments. And that's one of the reasons that I try to be careful between talking about neuroimmune modulation and neuroinflammation. After a while, I probably use them interchangeably. But when I initially start, I try to be mindful of separating those two. But I definitely agree that what we're seeing, particularly as it relates to the drink in the dark and those early non-dependent parameters, I would definitely say that it's probably much more of a neuroimmune modulator than being associated directly with damage or any kind of true neuroinflammatory response. Because we have actually gone back and looked at blood-brain barrier disruption in both models, and there's no blood-brain barrier disruption, so you're not getting those peripheral immune responders in the drink in the dark model.
Well, that's great. I had another question. There's a phrase, falling down drunk. And so sometimes people get drunk, often when they first start drinking as teenagers, and they have traumatic injury to their brain because they hit it on something. So as a converging risk factors in terms of the kind of immune response to kind of this neural trauma, might that kind of converge with the intoxication and withdrawal from intoxication to cause further damage?
So yes, there's been some work looking at TBI and alcohol as it relates to this. And one of the terms that I usually talk about in a different talk is the idea of the priming response, which Stacy Bilbo likes too. So the microglia and astrocytes, maybe less so than the microglia, as it relates to these models, when it persists out so long, any future immune event can actually have a greater response. And so there's been a lot of work showing that pre-exposure to alcohol can exacerbate TBIs. And the other direction, it's not as strong. So they've done TBIs while drunk and actually seen differing responses because and one of the reasons for that is during intoxication, especially in clinical tissue, you actually get a slight depression in some of the immune responses peripherally, not necessarily in the CNS. But that peripheral immune response may actually help mediate, or that depression may actually help, I guess, alleviate some of the brain damage when you're actually drunk and driving. Not that I say that you should be drunk while driving in order to decrease your TBI, because the drinking is what leads to the accident. But there have been some studies that have definitely shown stuff like that. But overall, I think the predisposition to alcohol does exacerbate those kinds of responses to TBI.
And then just as a follow-up in terms of the converging risk factors that, it's been shown, well, Fulton and others have shown this kind of excitotoxic damage with the recovery or withdrawal after the bout of intoxication. And so how does that excitotoxic cascade then interact with these kind of neuroimmune reactions?
It's been particularly important for when you think about the alcohol-dependent models and or clinically when you have disruptions to the blood-brain barrier. So there have been a lot of talks about how, gut leakiness or peripheral immune responses can actually increase the damage associated with alcohol. But I do think, again, that is dependent upon both players, both the central and peripheral immune response being activated. And so good or bad, when you are binge drinking, it seems to only be the neuroimmune response.
Okay, thanks. Other questions, comments? I had a final question about what's wrong with addiction? As a term. Why is George Krupp so dead set?
I know. I should probably just push through it, but we went through this whole seminar where we were told that we should not use those terms anymore, and they even put out a paper about it. So I don't know if you guys have seen it, but NIAAA in particular, I joke, but NIAAA has been talking about the stigma associated with those words and being more intentional about the phrasing that we use as it relates to people with an alcohol use disorder.
Yeah, well, having been around for a while, I think that replacement term will also acquire stigma as time goes on.
Yeah. And it's harder to say, right? Like
AUD, I guess. So,
yeah.
I think we should remove the stigma, not change the
terms. Yeah.
And that doesn't make any sense.
Yeah.
Changing hearts and minds instead of rules.
Yeah. That's a great thing. Well, thanks so much, Alex, for a wonderful talk and also a very interesting discussion. And value you as a colleague and look forward to our future collaborations.
All right. Okay. Thanks, guys.
Well, thanks all, and happy Thanksgiving. And I'll see you again on the 1st of December with Andrew Hockey.